Somewhere on the ocean floor right now, there's a whale skeleton with white bacterial mats growing across it, surrounded by animals that exist almost nowhere else on Earth. And the thing keeping all of it alive is a poison gas. I want to tell you about that, and then I want to tell you why it connects to something happening inside your cells at this exact moment. Because the connection is more direct than you'd expect, and there's a trap in it that I want to walk you through carefully. Let's start at the bottom of the sea. When a large whale dies, its body sinks. In the deep ocean, food is scarce, so a carcass that size is an enormous windfall, and it gets used in stages. First come the scavengers. Hagfish, sleeper sharks, crustaceans. They strip the soft tissue, and depending on the size of the animal, the bones can be picked clean within months. Then come the opportunists. Worms and mollusks that feed on the enriched sediment and on the bones themselves. And then comes the strange part. The stage that lasts the longest and looks least like anything you'd expect. Whale bones are not like our bones. They're loaded with fat. Lipids can account for around 60% of the weight of a large whale's bones. So even after everything else is gone, there's a huge energy reserve locked inside the skeleton. Anaerobic bacteria get into that. They break down those lipids without using oxygen, and as a byproduct, they release hydrogen sulfide. If you've ever smelled a rotten egg, you know hydrogen sulfide. It's toxic. In high concentrations around those bones, it reaches levels that would kill most marine animals. But another group of bacteria treat it as food. They oxidize the sulfide, and they use the energy from that reaction to build organic matter, the same job photosynthesis does up in the sunlight. This is chemosynthesis. Life running on chemistry instead of light. Those bacteria form white mats across the bones, described as looking like shag carpet. And on top of them sits an entire food web. Limpets, snails, mussels, clams, specialized worms. Researchers have documented up to 190 species at a single whale fall in this stage, and a substantial number of them are found nowhere else in the world. This can go on for decades. A large whale can support that community for 50 to 100 years. In many cases, longer than the animal was alive. Now, here's a line from a Scientific American article on whale falls that I want to hold up, because it sets up everything that follows. They wrote that animals cannot use this gas directly as a source of energy, and in fact the gas is typically poisonous to them, but certain chemosynthetic bacteria can. That's the standard framing. Bacteria can eat sulfide. Animals can't. And it turns out that's not quite true of you. Here's what's actually inside your mitochondria. Anchored in the inner mitochondrial membrane is an enzyme called sulfide quinone oxidoreductase. Sometimes shortened to SQOR. Its job is to take hydrogen sulfide, strip 2 electrons off it, and pass those electrons into the electron transport chain, the same chain that produces your ATP. Let me be clear about what that means. Hydrogen sulfide, at high concentrations, is a respiratory poison. It shuts down complex 4 of your electron transport chain, which is the enzyme I covered a while back in the video about light. So at a high dose it's genuinely dangerous. But at low concentrations, the same molecule is a fuel. One of the papers I looked at puts it exactly that way. In addition to being a respiratory toxin, hydrogen sulfide is also a respiratory substrate. Poison and fuel. Same molecule. Dose decides which. And this isn't an exotic emergency system. Your own body synthesizes small amounts of hydrogen sulfide on purpose, as a signaling molecule. Your gut bacteria produce more of it. So this enzyme is running continuously, keeping levels low enough to be safe and harvesting the electrons on the way through. Now here's the detail that puts this squarely in the territory I usually talk about. What accepts those electrons from that enzyme? Coenzyme Q. That's not a loose association. Coenzyme Q is the co-substrate. The enzyme oxidizes hydrogen sulfide and reduces coenzyme Q in the same reaction, and those electrons then travel on toward complexes 3 and 4. There's a further detail I find genuinely elegant. The coenzyme Q pool is shared. Several different fuel routes dump their electrons into it. And that creates competition. Remember the video about butyrate, the fuel your gut bacteria make from fiber? Butyrate oxidation feeds the same pool. And researchers found that when sulfide levels rise sharply, the sulfide enzyme can actually produce a compound that inhibits butyrate oxidation, freeing up space in the pool so sulfide gets processed first. That makes sense as a priority system. Butyrate is fuel. Sulfide is fuel that's also dangerous if it accumulates. So clearing the dangerous one takes precedence. Alright. Now I need to slow down, because we've arrived at exactly the kind of moment where this niche usually goes wrong. I promote a supplement whose main ingredients include CoQ10. And I've just spent several minutes explaining that coenzyme Q is the acceptor in a pathway that handles a toxic gas. And there's a study that fits that story almost too well. Researchers looked at what happens to sulfide handling when cells don't have enough coenzyme Q. In human skin cells lacking it, hydrogen sulfide oxidation was impaired, and the degree of impairment tracked how much coenzyme Q was left. When they added coenzyme Q back in the dish, the problem was rescued. They saw related effects in mice too, with sulfide accumulating in the affected tissue. If I wanted to sell you something today, that's the study I'd lead with. So let me tell you what it actually is. Those were models of a rare genetic coenzyme Q deficiency. The mice had roughly 15% of normal coenzyme Q levels and were clinically affected. In the same animals, brain tissue that retained around 30% of normal levels wasn't affected at all. That's a disease state. It is not the state you're in. And this is one of the most common tricks in supplement marketing, so it's worth naming clearly. Demonstrating that correcting a severe deficiency restores a function tells you almost nothing about what happens when you add more to someone who isn't deficient. Those are different questions with different answers. Scurvy is the classic example. Vitamin C cures scurvy dramatically. That doesn't mean extra vitamin C does anything comparable for a person who eats fruit. So I'm not going to use that study. It's interesting biochemistry and it's the wrong evidence for the claim it seems to support. What I'll say honestly is narrower. Coenzyme Q is a hub. Multiple fuel routes converge on it, including this sulfide pathway, and CoQ10 levels do tend to decline as we age. That's it. That's the honest version. Now the free lever, and I'll be equally careful here. Nobody has run a study asking whether exercise improves your sulfide handling specifically. If I told you it did, I'd be inventing it. What has been shown, repeatedly, is that training raises the activity of the enzymes in the electron transport chain substantially. I covered a study a few days ago where 20 weeks of running raised cytochrome C oxidase activity by over 40%. That's the machinery downstream of the pool we've been discussing. So the honest statement is that movement improves the chain generally, and sulfide oxidation happens to feed into that chain. That's an inference, not a finding, and I'm labeling it as one. Move anyway. It's free, and it's the intervention that keeps holding up no matter which angle I come at this from. And if you want to support the machinery itself, the link in the description below is an affiliate link to Mitochondrial, which is PQQ and CoQ10, with a 90-day money-back guarantee. If you appreciate a channel that walks you up to a study that would sell you something and then explains why it doesn't count, subscribe. That's the whole idea. This video is educational only and it's not medical advice. Talk to your doctor before starting anything new.